Let's look at the specimen jar on your bench and trace it to the microscope slide. That physical journey requires a chain of custody form and careful bench-level documentation. We aren't just processing routine surgical blocks here. Every label, transfer, and handoff must be meticulously tracked to ensure legal defensibility. Keep your focus sharp as we dive into the details. This slide contrasts two panels. On the left, the clinical laboratory jar is labeled quality problem. On the right, the courtroom jar is labeled legal problem. The contrast is not meant to say that every forensic laboratory error automatically ends a case, but rather that technical defects carry heightened consequences under outside review. An improperly processed clinical specimen is a quality failure requiring repeat work. But if the same failure occurs in a forensic case, the question shifts from whether we can repeat the stain to whether the evidence can still be trusted. Integrity problems can reduce the weight of evidence, trigger review, or leave a critical question unresolved. Everyone involved is accountable. Confession is never concealment. Read the chain from left to right because every visible link represents a different point of risk, from autopsy and accessioning through processing and storage. A chain is only useful if every handoff is traceable. It is not enough for the first and last labels to match. During accessioning, the laboratory verifies that containers and requisitions agree. During processing, cassettes must remain identifiable as they move through reagents. Every transfer must be recorded to ensure absolute confidence in origin. If an identifier conflicts or a transfer cannot be explained, we stop the process, notify the designated supervisor, and document the resolution. Never repair a broken link by just guessing. Examine the death to fixation branch diagram on this slide. Notice how autolysis and putrefaction split away from preserved tissue when delay occurs. Autolysis means self-digestion by enzymes, while putrefaction involves bacterial decay. But prompt fixation can still preserve remaining cellular details if we act fast. That timely intervention is your most critical tool at the bench. Let's get fixation right by following the core bench parameters. Always use 10% neutral buffered formalin with an adequate volume ratio. Keep your tissue slices at 3 to 4 millimeters for proper penetration. Allow 24 to 48 hours for thorough teaching benchmark stabilization. And document every step of this process in your batch logs. Let us examine how processing, embedding, and sectioning can make or break our microscopic analysis. Overstaining yields muddy sections where crucial nuclear and cytoplasmic details vanish into the background. Understanding leaves tissues too pale Understaining leaves tissues too pale for subtle pathologic features to emerge. Stain precipitant creates distracting specks that mimic microorganisms or cellular debris. Air bubbles trapped beneath the cover slip distort the optical field and hide critical diagnostic zones. Water artifacts introduce cloudy distortions that destroy visual clarity. Catching these flaws during quality control requires asking if the defect compromises the forensic question. If recutting is impossible, you must honestly communicate the limitation rather than pretending the slide is pristine. And this precision directly protects the integrity of our legal and ethical framework. Objectivity means we evaluate every forensic block without bias or outside pressure. Confidentiality ensures sensitive case details remain protected within secure lab channels. Documentation requires writing down every technical action and deviation as it happens. Dignity reminds us to treat every human tissue sample with absolute respect. Clinical excellence alone does not automatically guarantee a forensically defensible result. While clinical standards focus on patient treatment, forensic work demands total traceability and accountability under intense legal scrutiny. Poor quality can obscure critical evidence, block meaningful peer review, or force pathologists to issue qualified conclusions. You must constantly ask whether a slide is technically sufficient to support a definitive forensic finding. If the target area is compromised or unverified, stop and address the limitation immediately. Every single decision at your bench directly influences how evidence is weighed in a courtroom. And that bridges us right into understanding your unique daily role in the laboratory. Your daily work sits directly at the intersection of three demanding professional roles. As a scientist, you apply deep technical knowledge to interpret tissue changes and spot potential artifacts. As a technician, you execute standard operating procedures with unwavering precision and reliability. And as a guardian of evidence, you maintain strict chain of custody and protect the physical integrity of every sample. None of these three dimensions can stand alone in a defensible forensic operation. You bridge the gap between the autopsy table and the pathologist's microscope. And that foundational responsibility carries us forward into our concluding insights. Let's wrap up this first week by looking at the chalkboard summary before us. Across our three-part sequence, Mini Lecture 1 covered the scope and limits of forensic histopathology. Mini Lecture 2 covered the medico-legal setting and laboratory role. And Mini Lecture 3 covered tissue as evidence through preservation, quality, and ethics. And as we close, let's preview where we go next. In Chapter 2, we will tackle specialized stains, controls, and advanced artifact analysis. Remember that every advanced technique still depends on your basic bench discipline. See you next week.